How about the recording process? I think now it started recording.
Yeah.
Oh, Praveen sir.
Okay, sir. Sir, please. Yes, sir. Yes, sir. Please, sir.
Yes, sir. Yes, sir. Yes, sir. Okay, sir. Thank you, sir. Yeah, now I think I can start my session. Good morning to everyone. Welcome to the session one on target discovery, especially on introduction to biological databases in this 15 days online certificate course on artificial intelligence and machine learning approaches of drug and vaccine design in cancer. Okay, so some brief introduction about
Sir, I am.
me. Yes, sir. Or you tell me, sir. Professor Praveen, sir. Anything you want to convey? Yeah, I'm Dr. Praveen Kumar PK, a professor from Department of Biotechnology, Sri Venkateswara College of Engineering, and I'm also working as a Tare Research Fellow in IIT Madras. Okay, of course, I'm a PhD supervisor in Anna University. And about this certificate courses already conducted from Institute of Innovations, which is approved by MSME. So I just started delivering the certificate courses from January 2021 onwards. Like actually on the odd semester 2020, that is during the pandemic times. So we have also uh, participated a drug discovery hackathon event, a national event, and we have cracked nearly 3.6 lakhs internship. So based on that kind of a kind of an achievement, I think we just started um, training students on this drug and vaccine design courses. So if you see this certificate courses, the initiation taken by Professor Praveen Ramanachan sir. So first certificate course was happened on 16 to 30th January 2021. Later we had a few courses also, even the SVC, Department of Biotechnology, SVC in association with IIT Madras. And excuse me, the participants has to mute. Yeah, so we also conducted a kind of a certificate course, 15-day certificate course approved by Controller of Examinations SVCE for, I think it's a 1 credit course in SVC. Later, all the courses, whatever I trained is for the Institute of Innovations, MSME. So what kind of courses I have trained is basically on computer-aided drug and vaccine design. And recently, like 6 months before, I just trained on system biology approaches of drug discovery in cancer. And now I'm going to train in this topic. So earlier, I also trained specifically for drug design for SARS-CoV-2. So based on the achievement in drug discovery hackathon, we framed a certificate course, and I just trained accordingly. Okay. So more about this, why I have to focus on this specific topic, that is artificial intelligence and drug design. I mean, machine learning approaches of drug and vaccine design. Why we have to focus? Because from 2020, many people turned towards this the biotechnology era in development of vaccines, especially SARS-CoV-2 vaccine. So when so many people are dying, and not only the epidemic has turned to a pandemic situation, so many people are in search of the drugs and vaccines at that age, isn't it? So 2020 to 2030, I think we believe we can find more funding anticipated, not only from the national bodies, also from the international organizations and the bodies, okay, NGOs. And if you see this kind of, and in the next presentation, that is on tomorrow presentation, I can tell you what are the courses I've conducted in the Department of Biotechnology SVC. Actually, like for the past 2 years, I have received grants from AACT, CSIR, Department of Biotechnology, recently AACT, to conduct an Atal FTPs. Almost, I got grants from all the government organizations, okay, especially in drug design in cancer. So just because of we are getting funding not only for conducting the kind of a training programs like workshops, FTPs, seminars. We also conducted popular lectures in biotechnology from DBT. So we also received project grants from DST. We also anticipate a few more project grants from this, especially in the area of drug design in cancer. Okay, and also whatever I'm working in IIT Madras, I'm focusing on cancer research, especially in protein bioinformatics. So I'm working under India's #1 bioinformatician, Professor Michael Komiha in IIT Madras. So we are also uh, 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 in his lab, he has developed more than 20 to 25 different tools for proteomics research in cancer. Even myself, I already involved to develop one or two tools, okay, for the cancer research. So now, if you see this first chapter about biological databases, majorly in cancer, we should have a brief introduction on what are the different oncology databases available. I think if you see the biological databases, you have so many textbooks. You find a data on various datas and various databases available publicly. But in this kind of training, the certificate course, we should have an idea, especially on the oncology databases. You see, in my research, I'm majorly focusing on hepatocellular carcinoma, which is a primary liver cancer. So, about this kind of a hepatocellular carcinoma, I just fetched the kind of extracts what I found from my research in the and drafted in the form of a PPT. So I just focusing on hepatocellular carcinoma in most of the slides. So if you see the significant genes in hepatocellular carcinoma, you can able to browse through mycancergenome.org. In mycancergenome.org, just search for hepatocellular carcinoma. You will be getting so many genes. Okay, if you click this plus, obviously that in AKT1, how much percentage of gene is expressed in hepatocellular carcinoma, you will be getting that kind of a details. You know, whatever the data available in this mycancergenome.org is the data from the microarrays and from other wet lab techniques. Whatever techniques they have utilized for evaluating clinical trials in humans, that kind of a data, that the gene expression data is available in this databases. The first database what I've shown is mycancergenome.org, which is common to all the types of cancer. I just search for hepatocellular carcinoma and I've shown a page. And similarly, if you want to specifically go to the hepatocellular carcinoma kind of a genes information and the other kind of data, you can able to find from predictor.nchu.edu. This is from Taiwan. Okay, it's a database for hepatocellular carcinoma. Even there is one more databases, especially for carcinoma, oncodb.hcc. Okay, if you default type in Google, it directs to you this kind of a website. Okay, so here you can find what kind of experiments are done, how the data is deposited, and more information on the protein and how we can link with the Swiss Prot ID, a universal ID. So all these things information you can find in this oncodb.hcc. And there is one more database called the from Raghava, that is Institute of Information Technology, I2IT, Delhi, okay, which has a Raghava scientist has developed a cancer liver database. So where you can find the data is not only for the cancer, but you also find for the from the normal patients. So if you like to compare the gene expression of the normal versus cancer, you can you can use this kind of database to find more information. Because if you are stepping in onto mining the data of a cancer, a specific type of a cancer, so we can explore different biological databases to get a kind of more validating results. Okay. So because in artificial intelligence and data science, especially in AI, we should develop a precise training data set with more data, isn't it? So all these databases give you a kind of a clear idea about how to what kind of data we can fetch from what kind of biological, especially oncology databases, and then we can proceed upward. And now I take you to the major portal called C-Bio portal. This is the C-Bio portal is from the cancer genome database. Okay, in this cancer genome database, you find more information. This is a fully annotated kind of a database for, again, specific type of a cancer. Okay, so if you see a liver cancer, more than 97% of the liver cancer is hepatocellular carcinoma, but we have nearly other 99 types of liver cancers also. We have subtypes in liver cancer other than hepatocellular carcinoma, but major people with liver cancer has these kind of HCC symptoms. Okay, the HCC is one of the fifth stage of cancer, final stage of liver cancer. And if you just see this kind of data, what data we can find? Okay, so most of the statistic there, the NGS data, next generation sequencing data, are available in this kind of the C-Bio portal, especially if you want to see the molecular profiling related to methylation, mRNA expression, mutations. So, the hepatocellular carcinoma is not just caused because of the habits, also it's caused because of the virus, isn't it? Can we have a kind of interaction run? I'm focusing on a particular type of a cancer, isn't it? So can I want to check how much you have a knowledge on that. So I just type a question in the chat box. You can able to answer for this. Like abo- so I was explaining you something about the cancer. So I think nearly 229 participants are joined at present. I can't hear your voice. Now I am audible to you or not? Hello, participants anyone can interact.
Audible, sir.
Yes, yes,
Yes,
yeah, yeah.
you
I
are
think.
perfectly audible.
Oh, thank you. I think some internet connection problem with the participant. Now I think I can proceed. So the question is like uh, related to cancer. Yeah. So actually, the hepatocellular carcinoma. Okay. Uh, we have nearly 5 stages of cancer, isn't it? So what stage of cancer? What stage of liver cancer is hepatocellular carcinoma? Yes, very good. So I think you are following now. I think like this, you can have a kind of interaction because I want to know how much you are following. Okay, so more about the mutations. Okay, and yeah, I also want to ask one more question. What is the major source, major cause for HCC? What is the major cause for HCC? Anyone have an idea on that? Because just learning the computational information, computational biology for cancer. Yes, very good. The first answer is virus. The virus is number one. So especially hepatitis B virus and hepatitis C virus. These are the major, nearly more than 70% of the cause for hepatocellular carcinoma is the virus. The next is the habits. What kind of habits? Smoking, drinking alcohol. These are the other causes of the cancer. Okay, but if you see this kind of a cancer, okay, this cancer also leads to various mutations. So more about the mutations, the mutation data can also be fetched from C-Bio portal. Okay, and if you see related to the genetic disorders because of genetic the genes carried out to the successful ancestors, so you can able to find even the cancer is originated from the ancestors, isn't it? So because of hereditary and genetic disorders, so related to hereditary and genetic disorders for a specific type of cancer, including HCC, we can search for in online Mendelian inheritance in man database. Okay, so even in NCBI, we have this kind of a database, or even you can search in OMIM related to gene specific or hereditary specific disorders of HCC. And there is one more database called Genomic Data Commons Data Portal. This is also a large database. Okay, if you see the database, generally in biological database, we classify as primary, secondary, and composite, etc. Isn't it? So if you see these kind of a primary databases, major information, one I can tell CBio portal, which is common to all the types of cancer. Similarly, this is maintained by National Cancer Institute, of course, from National Institute of Health. This is the largest database, of course, GDC data portal, where you can find from the 86,394 cases, the data are deposited for almost all the types of cancer. If you see the cancer, or how we can tell a cancer, a patient is suffering from infected with cancer, we can tell from the abnormal tissue find in a specific site, isn't it? So if you see in face itself, okay, we have tongue cancer, we have tooth cancer, we have all the wherever you find organs, okay, so in all the organs we can find the cancers. So nose cancer, eye cancer, all the organs have the can infected with cancer. So if you see the types of cancer, and we can also not only see uh, the cancer, the type of cancer with respect to origin, like for example in US. So in US, because this portal is maintained in US, isn't it? So if you want to see what are the major population of US infected with cancer, you can find the lung cancer is most. Okay, you can also find more information which is updated on daily basis in Genomic Data Common portal. Nearly 72 projects, 67 primary sites, this much cases, files, genes, mutations. See, this much mutations are reported, 26 lakh mutations are reported. Okay, and related to the URL is mentioned below. So GEO gene expression database. So how these data are actually deposited? Generally, these data are deposited from the clinical data derived from the hospitals given by the doctors and other clinicians. And if you see the kind of other portal, that is gene expression omnibus. So majorly, if you see 10 or 15 years before, most of the data of cancer, okay, or gene expression data, okay, the gene expression data are generated from the microarray, isn't it? So microarray, but nowadays the gene expression data are generated from the RT-PCR. Okay, now if you see this kind of a gene expression kind of a data, okay, so even they will be mentioning the types of microarrays, what they've used to generate these kind of a data sets. So there are actually types of microarray chips and types of microarrays. All this information, or even the type of technology also mentioned in this kind of GEO database. Okay, so for a particular protein and what kind of a technique and how many samples they have taken, these informations you can find. So differential gene expression in from an NCBI web resource. And there is one more, this is a specific kind of a website that is from a Chinese database, the TCMID, Traditional Chinese Medicine Information Database, where if you can find the genes information for a specific type of a cancer. If you find a cancer, you can find the most important genes in that portal. There is one more portal called Traditional Chinese Medicine Systems Pharmacology Database, where if you just paste the SMILES format, or if you type the chemical name, okay, so there are different ways of mining the data in this database. So if you put any of the SMILES entry, or if you put the kind of a structure, or even if you type a chemical name, obviously you will be getting the details, the pharmacokinetic properties, especially drug likeness properties. All these properties are mentioned in this database. Okay, this is a kind of a specific kind of a database from a country. And if you see gene cards, this is also a primary database, primary biology database. If you don't know anything about genes, you have plenty of thousands of genes for a specific carcinoma. See, if I type hepatocellular carcinoma, how many results I have got? Nearly 8,100 genes are responsible for causing this hepatocellular carcinoma. If you want to find more description about the genes, even some few genes are not present in few databases, but this is the largest collection. If you want to find information on any gene, you can able to find from this gene cards. So genecards.org, you can find like this. So you can type a keyword or a type of a cancer and you can search upon it. And now if you see the next database is related to proteome. So if you see the kind of bioinformatics kind of a research, computational biology research, can anyone tell me when the human genome is drafted? My next question is, what is the year the human genome was drafted? Because if you want to diagnose the cancer, if only if you identify the gene, then only we can able to uh, treat a particular cancer, isn't it? For cancer, we need precision medicine. Very good. I got first answer from China. Actually, the answer is 2003. They have drafted in 2001, but for submission, it took 2003. The human genome submission happened on 2003. So in the 2000, nearly, if you see the mice and rat genomes published during this region, 2001, 2003, 2002, something, all this happened in this 2000 to 2005 era. And even 1000 genome project came in the 2010 itself, okay, before 2010. Like if you want to get a precise data, not from a single patient, but if you want to get from nearly 1000 people, so we also have 1000 genome projects, not only for Homo sapiens, but also for the other species, even for Oryza sativa, for Pisum sativum, for all these things, we have 1000 genome projects, where we are getting uh, a, a trained set with 1000 data so that whatever information you want to fetch, it will be in precise. Okay. So 1000 genome projects. So if you see the genome kind of era, so even the 1000 genomes came in the 2010, so before 2010. Then what is happening in the latest research? The proteome came into existence. So if genomes are established, then obviously, because of sufficient genes and genomes information, they also focused on proteomes later. So if you see in 2010 to 2020, many proteome projects are established. Okay. So the protein-protein interactions are important to design a precise medicine for a particular patient who infected with a particular cancer. So nowadays they won't if we are suffering from fever, obviously the doctor give paracetamol, isn't it? But if a patient is suffering from hepatocellular carcinoma, that doesn't mean that a sorafenib, a marketing drug for this cancer, is won't cure for all the patients, for all the HCC patients. Nearly 1 in 7 patients can be cured with the FDA approved kind of a drug. Okay, so whatever the standard drug, if you see in Chennai hospitals, I just inquired nearly 3 hospitals, the major hospitals for this liver cancer. They are majorly giving the syrup, but it's not going to cure. Only 1 in 7 or 3 in 7 based on the genes. Okay, if and if the major cause for hepatocellular carcinoma is VEGFR or EGFR, if these are the primary targeting receptors, then the kind of a drug sorafenib can able to cure. But if that is not the major gene, then obviously you have to look through the particular marker protein and what are the interacting proteins. So for this, there is a need to know more about the proteomes for the cancer. So the major the cancer proteome atlas gives you a network of proteins linked with a hub, a major protein what you're focusing on your research. Okay, so the TCPA portal. And you also have a types of networks. Here defaultly we have selected bind, but we also have few more kind of networks. We can select that and we can find how the proteins are interconnected in this network. Here I just search for the CCLE liver. We also have the other types of liver to search in the proteome atlas. And here in this slide, we can able to see the open targets kind of a database where you can find, if you focus on a particular kind of a protein target, you can type the particular protein target and you can find more information on the mutants available for the particular cancer and how the data, what are the data and how many clinical samples are taken. Okay, so based on the microarray data, of course, we find this information. And this is one such pathway database, KEGG pathway. KEGG stands for Kyoto Encyclopedia of Genes and Genomes. In this database, we can able to find the network of or the genes causing the particular cancer. Now, as I search for hepatocellular carcinoma, you can find there are so many pathways. There are so many, that not just a single pathway is uh, is the reason for the causing the HCC. There are so many pathways, plenty of pathways, nearly 7 to 8 pathways are established. And in my research, I am focusing on ninth, eighth, or ninth, however we call, which is unexplored. So I am focusing on the kind of the pathway which can able to treat the cancer, of course, with a kind of a precise medicine therapy. Okay, there are also, we also have an established kind of a pathways which we can find in medical textbooks. Okay, 8 different pathways. So each and every pathway, what are the proteins or what are the genes responsible and what kind of effects uh, the HCC patient face. All this information is available in this pathway database. So even the genes information, expression, all this also we can find in hyperlinks in that pathway database. That, of course, this is the largest pathway database. Not only we can find for a cancer, but also we can find this pathway database for all the types of diseases. GPA server. The GPA server, here you can find more in genes expression information, and also you can find gene enrichment. Okay, so the gene expression, gene enrichment, comparing the normal and tumor samples, all this information are clearly mentioned. If you can able to find generally by typing the marker protein in the quick search, or you can go for differential gene expression and survival, and we have so many fields to search a particular protein and the expression for a specific type of a cancer. Enricher, this is majorly for a gene enrichment database. If you type a protein, a marker protein, and if you want to search the top 100 genes or top 50 genes or top, we have number also. We can select that. And we also select the kind of a method. Here I have selected gene co-occurrence matrix. I want to find what are the top 100 genes responsible for interacting with HSP90 AIML. For example, if I can able to find the HSP90 is the major gene which is identified in the prognosis itself, which you find more expression when compared to the normal patients, then a particular gene what I'm going to search. Okay, I should not target just this HSP90, but whatever drug or whatever gene therapy I'm going to take, it should target also the interconnecting genes of the specific marker protein. Okay, here in this case is HSP90. So I have to find the top 100 genes. So for this, I can go for enricher server. For this, the URL is mentioned below. Okay, of course, in system biology approach, we have to take care of not only for focusing on the network, fetching the networks for a specific marker hub protein, but we also have to focus on the top 100, and we have to screen the top 10. And even from the top 10, if you want to find the top 5, even you can screen like that. So we have screening of the most important protein interacting with the hub protein in the interaction networks. So we have one database called string database. This database we can find protein-protein interaction networks. So here almost 14,094 organisms are utilized to find 67.6 billion proteins in the interactions. If you see 20 billion interactions are there. They have taken this much number of proteins and the interactions are crossing more than 20 billion. And if you see in the string database, how the data are deposited, the data are deposited through various wet lab experiments. Okay, mostly immunotechnology experiments. Okay, so all this information also available in this kind of a string database. And if you see the string database, how a data looks like, this is my one of my target protein. This target protein is interlinked with so many proteins. So if you want to target the drug, I should not limit up to targeting a single protein, but I have to target also the most closely related proteins in a network. Then only I can tell whatever drug I take, it can able to cure, and how much ability it can cure. It's based on the binding affinity. Through computational studies, we can able to at least predict something which can be predicted within less time when compared to performing preclinical studies and clinical studies. And functional enrichments can also be is already that these kind of functional enrichment of genes or the functional enrichment of the protein interacting with other proteins are already deposited in the string database. So the gene ontology information related to biological process, molecular function, cellular component, these information are present. Not only that, we also find the interconnecting KEK pathways, reactome, okay, pathways, wiki pathways, tissue expression. So all this information is also available in string database. So if you see the proteome research, the breakthrough of protein-protein interaction start from the major database string. Okay, only from the proteomes kind of the protein bioinformatics or proteome kind of a thing start from this kind of a string database. So one should explore more information about a specific type of a cancer and also other types of diseases in this database. So one such snapshot is shown related to functional enrichments happen in string database. The Professor Praveen sir, are you available? KEK full form is Kyoto Encyclopedia. I can type that. Kyoto Encyclopedia of Genes and Genomes. My Encyclopedia of Genes and Genomes. Okay. So this is from Japan website. Okay. I think I can continue my session. I think presently is not available. Functional enrichments. What I mean by functional enrichments. So if you see a specific cancer, for example, you are focusing on one particular protein for EGFR. Suppose if you're focusing on epidermal growth factor receptor, then you can type on epidermal growth factor receptor. And then you have, if you're focusing on a liver cancer, especially hepatocellular carcinoma is a liver, isn't it? The major tissue expression we have to initially check in the liver. So other than that, it can also focus on the other parameters. So in tissue is majorly liver. And how about the other parameters? This all we can able to find when I take you to the hands-on experience in this portal, string database. Okay, so we have so many screening parameters. And we can construct the protein networks based on the screening parameters. That's what we call it as a functional enrichments. Okay, so more about this we'll be seeing the subsequent sessions. So functional enrichment, one thing has happened, like if you here in my case, I just focused on a protein called HSP90. In HSP90, there are 10 major interacting proteins connected with it. But if I focus on a specific liver tissue, only these 1, 2, 3, 4, 5, only these 5 proteins alone or more connected or showing a tissue expression, a kind of a protein-protein interaction with the HSP90. These are all now are working like hidden states. No, in hidden Markov model, you have heard about machine learning technique, hidden Markov model, where only the most significant are shown in the normal Markov states, whereas the remaining, which is not shown, which is not significant, is hidden, isn't it? So the string database works, the functional enrichment in string database works on similar kind of a machine learning technique, where only if you select the lever, so you can find the red color. Similarly, in the interacting proteins of the 10 important proteins, only the specific function, the liver tissue expression alone is shown in the functional enrichments. Similarly, even we can able to go for functional enrichments with the two parameters. Here, initially in the tissue, I selected liver, but if I find there is also one more filter called a type of cancer, I just selected hepatocellular carcinoma. So initially liver, next hepatocellular carcinoma. For a liver, now I got blue color. So, but we have found so many proteins. So initially I got only 10 proteins, but what I did is in the settings, I have increased count to 50. I want to find the 50 top interacting proteins with my hub protein. Then I got this much number of 50 proteins, and from the 50 protein in the protein-protein networks, I want to screen based on the functional enrichment. So what I did is I selected two parameters for screening. Number one is for the liver, which shown in blue color, and if it is specifically to the hepatocellular carcinoma, it might have shown in red color. So only the blue and red are significant to the specific one. But if you find the most significant proteins in the network I found from the expression, these are the 5 proteins which I found from the network for the specific protein what I searched. So even you can search, you can fix a protein target and you can search accordingly. There is also one more database called Stitch. Like how I mentioned string, there is also a database called Stitch. This Stitch database can find not only the protein-protein interaction networks, but you can able to find a drug-protein interaction networks. There is one drug called selexopyr and indomethacin. There are 2 drugs I have shown. Even you can search for a single drug, okay, and also you can find the interacting proteins with the drug. Like the drug protein binding is very important to determine the dosage of a drug, and how efficacy a drug is going to treat a disease with by targeting a specific, isn't it? For this, even you can utilize this kind of a Stitch database. So in Stitch database, not only you can find for a single ligand, for multiple ligands are also shown here. But generally we search for a single ligand. Here in a string, I think I've already shown that the protein-protein networks we generally find in the string database, the largest database for protein-protein interaction networks. So whatever networks you can find from the Stitch, okay, if you want to download the data, you can able to download the data from the database itself. And if you want to import this kind of a networks in the Cytoscape visualizer, you have to install the Cytoscape visualizer initially, which is a free software to visualize protein-protein interaction networks, gene-gene interaction networks, or it can be even gene regulation networks or whatever signal transduction networks can be visualized using this Cytoscape visualizer, which is a freeware. So initially you can download the freeware from the going to the home page of the Cytoscape. And after installation, if you want to directly import to the visualizer to visualize the proteins, and if you want to do the functional enrichment in the visualizer, you can make it out, because whatever data you fetch from the database is a copyrighted data. You can't able to use it for publications. If you want to use it for publication, better download the data and you can visualize in the Cytoscape and do the functional enrichment. Then you can use the data for the publications. Okay, so for all this, you need a kind of a visualizer. So one such data taken from the string database and visualized in the Cytoscape. When I click the Cytoscape visualizer in the home page or in the string database, this is a data taken from the online. So if I click it, so even through the offline, you can able to see this data. Okay, you can store the data, you can save the data, and you can do the kind of whatever enrichments you want to make it up. Okay, so this is a window of a cytoscape visualizer. So I can find the kind of interacting proteins information, the nodes information, edges information, other kind of detailed information of each and every nodes. Okay. Then something about MATLABs in biology. All these things we can see in the next session. So now I believe I just recap what I just completed. I just mentioned you about some of the certificate courses I already had in collaboration with Institute of Innovations MSME. So taken courses like completed drug and vaccine design and something a drug design for SARS-CoV-2 for 5 days. So the remaining all 15 days course and system biology approaches for drug discovery in cancer and recently artificial intelligence and machine learning approaches for drug as well as vaccine design. I started with a type of oncology database. Not only we focused on the gene databases, then we have gone for the protein databases, we have gone for a genome specific oncology databases, we have gone for gene expression databases, and for the hereditary kind of disorders of a specific cancer, we have gone for the OMIM. And there is also a region specific kind of a cancer database, for example, TCMID, Traditional Chinese Medicine Information Database. We also have TCMSP, that is Traditional Chinese Medicine System Pharmacology database. And we have also gone for gene cards information. Like if you don't know anything about genes, if you have plenty of genes information and some genes information not available, better fetch from this kind of a large gene database called gene cards. And if you want to start a proteomic kind of a research, start with the cancer proteome atlas and also the string DB, which is a protein-protein interaction network database. And if you want to focus a specific target for a cancer, you can focus on open targets information database. And if you want to focus on the pathways for a particular cancer, and then for this, you can focus on the cake pathway. Okay, so we can't able to do, take it all the proteins. We can't study all the proteins in vitro or in vivo, but we can. I mean, we can focus on one or two proteins. Okay, and we can explore more. For this, you need a cake pathway, which is an overall a kind of an atlas for the gene responsible for a particular cause of cancer, and the GPS where you can find the gene expression enricher for the gene enrichment, string for a protein-protein network, stitch for both compound protein interactions, etc. Okay, so then the next class will be discussing more about the target identification, modeling, and validation. If time is there, I can start explaining more about even the MATLAB tools available for working on cancer. Okay, the MATLAB, it's a commercial software. Okay, not everyone can able to afford. If you have a crack version, you can use it. Or even for a student version, I believe it comes around 69,000, something like that. So I have a kind of a license version. So if time exists, I can also tell you how the MATLAB can be used for working on models, cancer models. Okay. The PPT can't be shared because I have more confidential information related to my uh, sponsored research from DST. So this information can't be shared, but the videos can be shared. Okay, you can able to get the recorded videos from coordinator Professor Praveen Ramanujam sir. Okay, if you have any doubts, you can ask me because on the first day itself, I don't want to. Now itself, I think I have taken much information on oncology databases. My suggestion is don't limit up to whatever I teach. Whatever I teach and whatever you are acquiring the concepts at that time, everything is fine. But all are volatile unless you go for practice. So initially, whatever I've taken, okay, websites, yes, all these websites, or you can able to find from my recorded videos. You can halt the videos and you can. I believe I've shown everything with at least time frame of 10 seconds for each and every slide. So halt the videos and you can take all the URLs and you can practice it. Okay. So anything else to be discussed? Any technical doubts you have? Can you tell about the GP7314 HCC? Yes. So I think related to this, you can able to find in gene cards, or you can find in the specific HCC oncology DBHCC databases to know more about the GP7314. If you don't know anything, actually, the researchers or layman in knowing about a specific protein, isn't it? If you don't know anything about a protein, better even we can able to search from UniProt, the general biological database. You have heard about UniProt? If I want to find anything uh, in this, so you can go for UniProt. Okay, go to the UniProt database, that is Universal Protein Database. If you want to focus on a specific protein, I suggest you to go to this. Where is my? Yes, yeah. GP73 is your protein, isn't it? So type GP73, but it gives you the general information, not specific to the. Yeah, still I don't find GP73. Here I find something, but something related to Shigella page. If you want to specifically want to search for the human, we don't have information on this. So if you don't have information in the UniProt, which is a fully annotated protein database, then obviously you have to search in this gene cards. I told you, no, gene cards. In gene cards, you can search and you can explore the information. Okay. Anything else to be? Any other doubts? Difference between TCMSP and AdmetLab. Of course, TCMSP is a derivative. AdmetLab is the largest one. We find more information on AdmetLab. TCMSP, the Chinese people, whether you believe on the Chinese products, sometimes you go for Korean products or Japan products for quality reasons. Even the websites are like that. So instead of relying on a region-specific websites, we better go for the primary kind of oncology databases, which gives you a more better information. Okay. Even we may have some kind of errors, we don't know, but better go for the primary oncology databases. Anything else to be discussed? Am I clear? You said many database, but if you show us how to work on the database, it will be better. For example, how you showed us a UniProt database. Yeah. So actually, I can able to show, but this we can able to see in the subsequent classes. In subsequent classes, anyhow, if I want to explain on a specific tool, I have to take you to the database and I have to download the data and I have to explain, isn't it? So I can able to take it accordingly. Okay, if you want to go for the specific, because of one, at least one participant was specifically asking about the GP73. Can we go to the gene cards and I can fetch you some information on that GP73? Okay, then initially you have to go to the specific cancer and then you have to find accordingly. So this GP73 is alias name is GOLM1 gene. I think it's also available in UniProt. So from this I got a confident information. So I have to find like this. Yeah. So I can able to search, even you can order the products. Not only can find the information of a particular gene, but also find you can order the proteins from different vendors available the gene card. So of course, the largest gene database. So more about the information, additional gene information. So we have the gene expression information, transcript and factors. size, and other kind of a gene target for the specific genes. All this information are present, and what chromosome, in which chromosome you can find the specific gene. Okay, so you find the genome related information, proteome related information. So all these things are interlinked. We have so many hyperlinks for that. Okay, like however you find in UniProt is the largest protein annotation database. Even for genes, okay, you find this is the one of the largest database. Okay, anything else? So only thing is that you should have a zeal to learn. So in the first day, I think so many participants are interacted. I'm glad to find, because even if I want to take interesting, even the participants has to interact. Somehow the could you teach any pathways related to HCC? Pathways you find, even there was plenty of textbooks available for hepatocellular carcinoma. Okay, related to computational, related to clinical experiments. You can download from the EZ Lib. Okay, you heard about this EZ library, where you can able to download the free textbooks. You can download the textbooks and you can learn accordingly. So for learning about a specific cancer, you should focus on a specific cancer, and you have to download that kind of a textbooks. Okay, so you can start from the textbooks, and you can for contemporary researchers, you have to focus on the PubMed, focus on the recent years to download. Okay, anything else? Pathways related to, I told you, the KEG you can find, and you can also find uh, from journal papers. Yeah, anything else? If you have raised hands, you can even unmute and you can question me. If you can't, type in the chat box. One or two, you can unmute and question. Sir, is computational biology quite no? Actually, there are plenty of data publicly available in the internet, isn't it? So, and most of the tools are free. You know, if you take engineering, mechanical engineering, the 3D structure of alloys, okay, 2D structure are all commercial. If you want to download 2D or 3D structure, you have to pay $10, $15, $50 based on how much complex they have. But this is something related to our health. Anyone, all the people should know something about the health, and it should be publicly free, at least to prevent few diseases, to treat few diseases. So most of the biological data available in internet are free. So computational biology is easy to practice because we have free tools and free data available in the databases to work on. Okay, I think I've completed this today's session on biological databases, especially on oncology databases. So next class we can follow the target information. Have a great day. Good night to everyone. Bye.